Aposense, targeting new healthcare horizons. Apoptosis is a genetically controlled process by which nature eliminates unwanted or dysfunctional cells. If the cell suffers an insult, such that it cannot function normally, it activates an orderly systems shutdown, followed by fragmentation of the cell. The small fragments are then ingested by macrophages and eliminated without harming the surrounding tissue. The apoptotic death process is the same regardless of the apoptotic trigger or cell type. Being a fundamental process of cell biology, apoptosis is present in almost any medical disorder. Aposense developed a family of proprietary molecules that selectively target apoptotic cells. Illustrated here is ML-10, the lead compound for molecular imaging of apoptosis with PET, positron emission tomography. Attached to the tail of the ML-10 is the fluor-18 radioactive isotope, which is commonly used for PET scans. By tracking the isotope linked to the ML-10, the PET can create an image of the apoptotic areas in the body. At the head of the ML-10 is the proprietary nanoswitch, which enables its selective uptake by apoptotic cells. During IV administration, the nanoswitch is in its off position, and the ML-10 is in a highly soluble state, allowing it to quickly distribute in the body through the bloodstream. As it enters the extracellular space, it does not interact with normal healthy cells. But when approaching cells undergoing apoptosis, the nanoswitch responds to a set of unique alterations occurring in the apoptotic cell membrane and turns to the on position. The key membrane changes that are recognized by the nanoswitch are the acidification of the outer membrane leaflet and the depolarization of the membrane. In response to these membrane alterations, the nanoswitch changes its conformation and electrical charge. These conformation and charge changes increase the membrane affinity of the ML-10 probe and enable the molecule to cross the membrane and accumulate inside the apoptotic cell. As it accumulates in the apoptotic cells, the radioactive probe reports on their location, and the PET scanner generates an image that allows clinicians to map cell death in real time. The yellow-red region in these PET images shows the apoptotic brain damage detected as a result of ischemic cerebral stroke. In cancer, ML-10 allows tracking the actual biological effect of anti-cancer therapy, as shown in this image of a brain tumor, where the red and yellow regions show increased apoptosis levels in the tumor following radiation therapy. Aposense, targeting cell death in vivo in the living body for imaging, therapy, and new healthcare horizons.